get going just a little bit early, just into a little bit of housekeeping stuff. So today, has everyone here done genetics with Jenny Chaplin? Has anyone not done genetics, or they're doing it at the moment with Jen? Okay. So this lecture's moves slightly forward, so it's a little bit out of context compared to how we had it last year. But the idea is to give you some extra time to do the assignment, so you've got weeks and weeks to complete what you're going to do this afternoon. So we're kind of getting in a little bit early. So if you're a little bit daunted by the whole thing, don't worry too much. Hopefully this afternoon will explain a lot more about what I'm going to talk about today. Just to give it in context. Those Penguin Island books, so far I'm averaging better than last year, but I'm still 4 of them weren't handed back out of the 6. So if you've got one of those little Penguin Island books, if you're too embarrassed to give it to me, that's fine. Just put it in my pigeonhole in biological sciences. We use them every year, so obviously we recycle them. So if you wouldn't mind to return them, that'd be very nice. And so what I'm going to do today is to talk about conservation genetics, but really particularly the importance of how we apply it in conservation sort of settings. So I want to stay a little bit away from all the formulas and all the guff and talk more about how we can actually apply it to animal conservation or plant conservation. And part of that, I thought I'll bring some of this stuff. This is coming from our lab. So we have a whole lot of wildlife seizures that come through the lab, and we'll be asked to identify these. Most of them we can identify morphologically, so we can actually look at them and just sit there and say, well, we know what that actually is. But a lot of them, if they're pieces of ivory, chess pieces, or spoons, or something's been carved up, we'll need to extract DNA from those, and then we can use the DNA to identify what the species is, whether an elephant was in fact a buffalo, things like this. We've had court cases, well, the court, and these things have been buffalo born. There's nothing wrong with buffalo born if you're in the buffalo born. But if it's elephant, it's a different story. You've got a protected species. So I thought I'd just chuck around a few different things, and there's sort of soups and stews and leopard plasters and things, and then at the end of it, maybe if we've got some time, we'll have a guess competition about what they might be. So hopefully you can see these things. Some are big, some are small, some of them are very expensive, others are not so. And the other thing is, I thought it may be worth mentioning, this is not an official university announcement or anything like this, but obviously this coronavirus thing is setting off and triggering everyone. They shut down University of Central Queensland, I think, for a day or two. So universities are starting to shut down, workplaces are starting to shut down, whatever. I just thought I'd get in early. As I say, this is not a university announcement, it's more just to sort of tell you what I think we'll try to do with this unit if it does shut down. If things like field trips, so these are the things where when you've got lots of people in one room, they're likely to say you can't do that anymore because people are going to be spreading this virus around. So field trips are likely to suffer, or may do. You'll be able to do all of it online, so even the questions we give you, you can follow up, so do the virtual, not quite the same thing. Having said that, I figure once this thing blows over, because it will eventually leave and we'll get the next pandemic through, is we'll try and organise an alternate trip to those places. So it might be next semester, it might be in the middle of the semester. We'll just give you the option of saying, look, we've been able to secure another date if you're interested in going to, say, a Variance or whatever, if they get called off. Even Perth Zoo, we might do it maybe later on, have a chat to Kate in the second semester when this thing goes off. Does that sound reasonable to everyone? I think it's unavoidable. I think it's going to come, and at some stage they're going to say, uni's going to shut down, no one can. And so we need to make contingencies to do that. Having said that, one other reason for doing that is to say that timetabling are very unlikely to pick up those changes. So in other words, they're not going to immediately say, yep, we'll reschedule your field trip for here. So don't look for the timetabling for those changes. What I would do is look for that where should we be document, and I'll make sure that we update that. And we may do some tweaking around. So as I said, when we have a field trip one week, it might actually be a seminar if we can get the seminars in. It's not going to disadvantage you, I'm just giving you a heads up. We'll try to rejig everything so it works out a little bit. Does that sound reasonable to everyone? So keep your eye on that document, that where should I be document, in terms of what we do afterwards. So today, as far as I'm aware, everything's fine. So we've got 2 workshops in the computer. You will either be doing a 1:30 workshop or a 4:30 workshop. They're on LMS, the lists of people in each of those groups. Does that sound, yep?
How long do they run for?
Workshops? I think they're a couple of hours. Yeah. And this one's important because there's an assignment, an assessment piece attached to that. So that won't be due for several weeks. Okay. And the only other thing I'll send around is this little gadget here. This is the internal of the human, in fact, fragment analysis. This is a DNA sequencing. So this won't mean a lot at the moment, but it will when we get up to the slides and thing. I'll chuck it around. But it's just to give you a bit of an appreciation of where the technology is. So This piece of apparatus sits inside a DNA sequencer. This is the thing which sequences the human genome, and not this particular piece. This thing's worth about $5,000, and they're a 16 capillary array, and each of those capillaries is hollow, and DNA moves through the capillaries and separates according to size. It will mean a lot more once you get there. I guess the appreciation is that DNA gets sucked up down this end. It moves up through these capillaries and separates into different fragments and gets detected by laser in that little window there that's etched out, and then it gets chucked up to the bin. So I'll chuck that around as well and have a look at that. Cool. Rightio. Let's get going with the lecture. Rightio. Everyone okay? Rightio. So today what I want to do is to talk about conservation genetics, and it's really an area of genetics called population genetics. And as I say, if you're doing this unit with Jenny Chaplin, later in her unit you'll pick this up, and this will hopefully mean a whole lot more when you actually do it. And if you've done Jen's unit, you will have covered this already, and hopefully it just puts it in a bit of perspective. Conservation genetics is to do with understanding the underlying genetic problems of what happens with populations when they go wrong. And if you think about it, your DNA forms all the things that involve your life, all the obvious sorts of things you've done in cell biology and things like this. But if you think about it, half of your DNA comes from your mother and the other half comes from your father. So there's a history to that DNA. Half of their DNA comes from their mother and their father, etc., etc. So at the end of the day, if you look at your DNA, you're actually looking at a historical map of what was happening in years gone by as a result of accumulating all those differences between those generations. So DNA holds a history, and it allows us to look back in time, not just at a snapshot today, or in fact it allows you to do that as well. And there are 11 major identified issues of genetics involved in conservation biology, and I'm going to go just through each of them quickly, but I want to go through a little bit more detail later on. There's the effects of inbreeding on reproduction and survival, and people may have heard of inbreeding depression. What happens is the population decreases to the extent that the only available mates you have in a population are highly related individuals. Because they're highly related, you will tend to accumulate all those inbred genes, and eventually you start losing the ability to reproduce, survivorship decreases, etc., etc. It's called inbreeding depression. We have loss of diversity, and this is one thing we're going to talk about a lot today, and this is underpinning what we're going to actually have in this assignment today as well. We have reduced fragmentation, or we have fragmentation populations, and as a result of that, we've got reduced gene flow between them. So what were really large populations, and Australia has inordinate numbers of examples of species that had continental distributions that have now been fragmented down into highly disjunct populations, separated by distances that just don't enable individuals to move between those populations. Whereas previously, there was a whole lot of movement within the populations because they were large and homogeneous. So the other result, or one of the other things that's a result of fragmentation is that we have random processes that happen because of small population size. And this is called genetic drift. And genetic drift is independent of adaptation. So in other words, being big or being the dominant male will no longer allow and give you any advantage over any other individual in the population. It's purely a random chance event that happens. So even a population, and these individuals survive and these individuals die off. Genetic drift is the movement of genes as a result of these small populations, and I'll talk about that a little bit later. Accumulation, purging of deleterious mutations. We get small populations, those populations purge out those deleterious effects very quickly because of size. We've got genetic adaptations for captivity. There's a whole lot of programs now in zoos, breeding programs, to try and give animals that they're releasing into the wild some sort of advantage for understanding things like predators. So we have numbats, and they release hundreds of numbats, and the same number that they release get eaten as soon as they get released into the wild by things like wedge-tailed eagles, large carpet pythons and things like this, because they're simply naive. So there's no adaptation, no genetic imprinting, I suppose, for the idea of a predator. So there's adaptation to genetics is really important. Resolving taxonomic uncertainties, I talked a little bit about this in my first lecture where I talked about if you don't understand what species you're dealing with, then it becomes very difficult to try and manage that. Imagine you're dealing with two cryptic species that have been identified as a single unit, and they're actually two different ones, then you're actually managing two different species under what you consider to be the same thing. So understanding taxonomic certainties, cryptic species, species that should be two species where there's one that should be split, really important. Defining units for management, again, I'll talk about that when I talk about some work we've done with feral pigs. And the use of molecular genetics in forensics. So this is a little bit to do with some of this stuff we're chucking around today. So some of that material has been used in court, and DNA and genetics form an important part of trying to identify what some of those things are from. And we'll talk about the genetics and the underlying molecular data this afternoon in the workshop. Okay, so what I want to do in this couple of lectures is a couple of things, and at a very broad level, I just want to explain what conservation genetics is. Hopefully you'll have a bit of an idea about that is. Why it's important, how we measure them, and these are the sorts of things that make good exam questions. What's this particular marker, and why is it really good for answering this particular question in conservation biology? How we measure genetic diversity in terms of the technology. I'm not trying to make you guys molecular biologists. What I'm trying to do is just sit there and explain there are different technologies around to answer different questions, and that's looking at that. And then, lastly, I want to talk a little bit about some of the things that change genetic diversity. Ultimately, what we want is we want to have populations that are maintaining genetic diversity, large amounts of genetic diversity, and those populations are changed when we have these sorts of things affecting the populations. When we have mutations, genetic drift, we have fragmentation effects that affect population size, etc. They change the way or the composition of those populations. And if we can understand what changes them, then we can mitigate in conservation management some of those changes and hopefully make and get them better. And then lastly, a few things on limitations of genetics in conservation, because it's not the answer for everything. Rightio. What we have in this planet we're living on at the moment is the result of 4 to 5 billion years worth of evolutionary change, accumulation, etc. And it takes a long time to have that genetic diversity in the population. When one of those populations disappears because of fragmentation and all the other things that we'll know about, you simply lose all that genetic information, and it's very difficult and probably impossible to get that genetic information back. So when continuous populations are fragmented into very small populations, the genetic diversity in each of them starts decaying, and it becomes an incredibly distorted system. So rather than a big random mating population, we have a series of dysfunctional small units that are genetically quite unique from one another. And maintaining those large interbreeding populations requires populations that are able to mix, and this is what I'm going to talk about with migration and all such. Go past that. So why is genetic variation important? Genetic variation is important because it's a description of a whole range of things. There is no such metric of genetic variation. There's a whole bunch of them. There are things like, and I'm going to talk about these later on, but things like heterozygosity and the number of alleles, the amount of genetic information, and also the mixture of those genetic things. And there's general consensus held that the more a population has in terms of genetic diversity, the more that population has the capacity to buffer change. So if you've got lots of diversity and lots of genetic variation in the population, if something affects that population, then you'll have the capacity for some individuals to survive, although maybe not all of them. Whereas if you don't, then you simply don't have that capacity for variation. So I've got a really, really simple example here. You've got a population that has individuals that may be cold adapted and hot adapted, and you've got another population that simply has no variation. They're all just cold adapted individuals. Global warming comes along, the planet becomes a little bit hotter. Those individuals that are cold adapted are not particularly adaptable, not particularly good individuals to survive in hotter climates, so they will become extinct. So what you end up having is that you'll have two populations, one which contains individuals that are adapted to an increasingly hot climate, and then you'll have a population that simply goes to extinction because it doesn't have capacity to buffer that change. So diversity allows at least some individuals to go through these things. And this may be all, you can think of it in a huge number of different things that go through there. things like susceptibility to a disease, variation for coping with the challenges of the different viruses, this thing that's going around at the moment. Why are some people dying and other people are not? Because there's variation in the system, there are some people that are able to cope with that and some people aren't. By far the most important thing we're interested in conservation biology is the forms of genetic variation that are for reproductive fitness. In other words, ultimately, we would really like to know how much a population has adapted in terms of how much height they've got, the weight, things like their survivorship, fecundity and things like that. How long will a population live for? How long will individuals in the population live for? How many young will they produce, etc., etc. But the reality is that for most of those quantitative characters, the ones we're really interested in, it's simply impossible to collect those in the time frame that we get. Often we're in a situation where we've got species. I'll give you a classic example was the woylie, where the woylie was put on the endangered list. It then went crazy with fox baiting, and numbers increased that they removed it from the endangered species list. It went to threatened and then vulnerable, so it even got higher. And then back in 1998 or so, the population for some reason crashed to almost 500 individuals. It was very, very small. So you've got a population that's absolutely been decimated. At that stage, there isn't capacity to sit there and say, well, why don't we work out how long woylies live for, or how many young they have per individual, etc. Because the population's at the stage where it's almost become a distinct sort of entity. So we need de facto methods of measuring genetic diversity or quantitative traits. And for that, we can use a whole lot of indirect methods. Rather than measuring how long an individual lives for, we can use their DNA to try and estimate how much diversity is in a population. And we can use them using a whole range of indirect methods. Things like, and I'm not really going to talk about the detail of these, you can go look them up if you want, but things like allozyme or protein polymorphism. So two different proteins in your body may actually have slight variation. We can look at DNA markers, and we're going to look specifically at these things called microsatellite markers. And we're now using these deep sequence things which require clouds and all that sort of stuff, but looking at entire genomes of individuals in a weekend. We can sequence numbat genomes in a couple of days now. Okay, so what I want to do is just talk a little bit about some of these some of these markers, these indirect markers. The first one's protein electrophoresis. We used to do this back when I first started working out in the field, and we had to go and collect animals, but the reality is, the problem is we had to collect various tissues of those animals, and some of them were not very nice. So essentially you had to euthanize an animal. You take samples from blood, from the liver, the heart, the kidneys, and then you'd go and homogenize these tissues up, run them on a gel, and then you would look for that variation in the proteins in those tissues. So you might be looking at a heart-specific protein in the heart, for instance, and you'd be looking for variation in there. In terms of an endangered species, that's not an ideal outcome to homogenizing every individual you capture. So it's a technique that has certainly gone out of phase now. But the problem is, it's only about 30% of the DNA substitutions in these proteins will give you any way of measuring genetic diversity. I'll give you an example of this in a second, where we've looked at the different techniques on the same individuals and how they vary in the information that they give you. So there are now techniques that will allow you to measure the DNA sequence directly rather than indirectly. So if you think about it, proteins are coded for by the DNA. So what you're looking at is DNA variation, but you're looking at it at the protein level. We can now look directly at the DNA and measure genetic diversity at the DNA level. And one of the things we can use is this thing called non-invasive techniques, which are things like polymerase chain reaction. Has anyone not heard of PCR? I've been teaching the unit for some time now, and it's really interesting how that decay of people who... So when I first started, most of the class had never heard of PCR, because it was a technique that was only just starting to come on, whereas now I think it's a standard thing, probably in high school biology, I would guess. It's great, yeah. So it's a technique that everyone, I mean, you may not actually understand the intricacies of it, but the gist of it is that you can start off with very, very small amounts of DNA and amplify up regions that you're interested in, and it allows you to then look at that piece of DNA that's been amplified and go, okay, how much genetic variation is it? I'll talk about the reason why it's important in a second. So what we understand about with DNA is DNA is made up of 4 base pairs, the AGCT, I think everyone can know that. And your genome is about 3 billion base pairs in size. Human genome is about 3.2 billion base pairs. So it's a really long piece of information, and it has a lot of information in it. But the reality is if we were to look at 2 unrelated individuals in the human population, 99.9% of that DNA is absolutely identical. So if I was to compare me with someone, one of you guys, then the DNA would be almost identical. Now that's of no use in conservation genetics for looking at diversity, because there's no diversity to compare us to. So it's this 0.1% difference that's really important in trying to understand genetic diversity and genetic conservation genetics. And these variations, or these what they refer to as polymorphisms, are really important. And they make us who we are, they make us unique. And they make us react to things very differently from one another. So the way I react to a particular disease will be very different than someone else in the room because of those genetic variations of polymorphisms in the genome. The way I react to an environmental insult like this coronavirus may be very different than other people in the room that may get very badly affected by it, or even worse, or any other bacteria and viruses, things like this, things like chemicals. And things like drugs and therapies. The way I react, or the way my body copes with something like paracetamol, might be very different than the way your body reacts to paracetamol. So you might find that aspirin's actually a better pain relief than paracetamol, you might find the other, etc., etc. So it's these variations, the polymorphisms, that allow us to cope with all these different changes. And what we're really doing is we're taking advantage of the fact that there's these little variations in the DNA to try and understand the diversity in populations. So in terms of molecular photocopying, or this PCR reaction, what it allows us to do is to take really, really small samples of something and amplify them up. So if we can identify those highly variable regions in the DNA, we can target them specifically with a PCR reaction and amplify up those variations, and then compare these populations and the differences between individuals in the populations. So we can use all sorts of material to start with. This is a study we did on the Madagascan giant bird. It's a ratite, similar to the emus, the mullahs out of New Zealand, things like this. So this will give you an idea of the size of this thing. It's a pretty big bird, it's extinct now. That's the size of the egg. The egg's about this big. And this is a pigeon egg or something or other, I think, or a quail or something like that, just for comparison. So it's a very big bit of egg, and there's lots of broken eggshells around the place. And we can actually extract DNA from the eggshells from these extinct things and look at what they are in terms of the species identification and things like that. This is a DNA gel. You may or may not have seen a DNA gel, but don't worry about the detail. I'm not going to ask you in an exam. Can you tell me the orientation of this and what is in lane number 59? Point of the slide is that the DNA goes up in these little things up here, and you put a charge against it. DNA moves against an electric charge, and you can separate the DNA. These are from 5,000-year-old human Bronze Age specimens out of Europe that I worked on in my postdoc, looking at transmission of HIV resistance in genes in that particular lab. And these are a whole series of individuals, and you can see that in most of them you can actually see the DNA. So the DNA is quite usable even after they've been sitting in caves for 5,000 years. Where does DNA come from? Can anyone think of a cell in the body where you would not be able to identify DNA?
Red blood cells.
Pretty good. So red blood cells. Red blood cells in mammals, not camels, not mammatrons, etc., not reptiles, not fish, but in mammals, don't have a nucleus. They expel the nucleus, and it's just simply an oxygen-carrying thing. So red blood cells, when they're mature, don't contain any DNA. Anything else? Anyone look at this list and think of anything that we wouldn't be able to get DNA from? We get them from everything. So any living material that's been in your body will contain DNA. And in fact, some of the best DNA we find, particularly in the ancient samples, in tusks and things like this, actually come out of things like the teeth or tusks, the big femur bones and things like this. Because if you think about it, when your teeth were developing, they were growing, there are these cementocytes, there are cells involved with developing the teeth. They develop the teeth, and once the tooth's developed, those cells stop functioning, but they still remain encased in all that enameling. So in fact, a lot of these individuals The tusks become the best material for DNA because it's the most preserved. It's the most difficult for things like funguses and bacteria to invade and break down. So we can get DNA from all these things. We were looking at parchments from the Middle Ages coming from goats and lamb and things like this. Jaws. We can get DNA from the ends of feathers, and we can sex the bird and things like that. This is an example of where I used PCR, and I don't know if I talked about this before. My PhD was on rock wallabies. These are a small 4 to 5 kilogram rock wallaby. The really interesting thing with these guys, it's called the allied rock wallaby. It occurs up in North Queensland. The weird thing about them, well, the intriguing thing about them is behaviourally they're monogamous, so they stay together for life. So they're called serial monogamous. So in other words, the pair will stay together while both pairs are alive. If one dies, they'll re-establish a pair and then that pair will continue. But they're very cool animals. They'll go out to a rock, they'll sit there on that rock pile that's exclusively theirs, and they'll allogroom, which means it's a bit like sort of chimpanzees pulling out fleas and things like this when they're mate. Everyone's seen footage of that. Rock wallabies do the same sort of thing. They feed together, they go over the rock together, they do everything together. So behaviourally they're monogamous, somewhat similar to humans. And we were really interested in knowing whether the pouch young from these rock wallabies were actually being fathered by the paired male, or at this time there were some really interesting studies, particularly in birds coming out, where they'd be looking at whether they were monogamous. So birds are predominantly monogamous, mammals are predominantly polygamous. In other words, there's a dominant male and subdominant female. So you have one male and a hell of a little sort of harem of females as a general mammalian pattern. So these are quite strange animals in terms of mammals from pair bonding. And they were finding, in fact, that there's lots of what they call extra pair copulations. So females were behaviourally monogamous, but they're actually sneaking off with some little on-the-side stuff and having young where you've got a clutch of eggs and there were multiple young in the eggs. We've just published a paper on northern poles, and northern poles can have up to 6 or 8 young, and we've got individual females where every single one of those young has been fathered by a different male, and they're all living in the same pouch. It's kind of a really, really cool pattern. But anyway, these things are monogamous. Poles aren't. Poles are naughty and don't do anything they like. And they're very good. So we're really interested in knowing whether these fathers were actually fathering those young, or whether in fact we had a situation like we did in birds where they sort of shopped around. And that's exactly what we found. If we looked at the reproductive success of a female rock wallaby, if the pouch young failed to survive, then the female would seek an alternate partner for the subsequent young. And if the female had a young that did survive, then she would mate with her paired male subsequent to this, when that pouch young was gone. So in other words, what was happening is that when females were being successful, they were staying with their mate, but when they weren't being successful, they were shopping around in the hope of getting something better. And these were usually larger males. So this is how you have this sexual dimorphism that happens in the big kangaroos. You would have seen the big male greys and the big male red kangaroos. They look like brick, they're huge. It's because there's selection for those larger males from females who are presumably being doing it. So that's what happens. I've got this table up in here simply just to give you a little bit of information. But the idea of the table is I'm not going to expect you, and I will not ask you in an exam, what is mitochondrial DNA sequencing, or what is AFLPs, etc., etc. But I guess what it's for is to just show you that there are these different genetic markers that are around, and they allow you to answer quite different questions in terms of conservation genetics. And by that what I mean is if we were to look at identifying individuals, parentage analysis, whether there's multiple fraternity, whether there's relatedness, whether there's dual samples, whether we're collecting 2 samples from the same individual. By far the best marker for looking at that sort of thing would be to use microsatellite markers. And we'll look at that today in this crack in the lab, so it should mean a little bit more when we actually do that. If we were looking at variation between populations, so we've got a population of rock wallabies and we've got another population of rock wallabies, microsatellites are good for looking at those differences, but there are also other markers that are equally useful for doing that. And then if we've got a very deeper question like, how related is that rock wallaby to grey kangaroos, then we're looking at these more phylogenetic or taxonomic questions. And by far the best markers for those are these mitochondrial markers. So the point of it is that there are different markers and they're answering quite different questions depending on the question you've got. And it's a question I get asked all the time in recovery team meetings. We want to answer this question, what do we do? And that'll direct you to what sort of technology you want to be doing. So I want us to talk about specifically a little bit more about microsatellites because they've been the foundation for genetic studies for about the last 20 years or so. And the reason that we use these particular markers is they're incredibly variable. Anyone following the Claremont killings at the moment? Because the DNA evidence in that has all been entirely based on what they refer to as microsatellite markers. So this is using exactly the same technology that has underpinned basic forensic science for many years now. Exactly the same markers, but they're obviously different. They're human microsatellites rather than kangaroo microsatellites or whatever. They're very high and show high levels of polymorphism. So at any one genetic marker, we can find lots and lots of variation. So it means it gives us lots of information on comparing things, whether they be individuals or populations. They're very short fragments. They're only a couple of hundred base pairs of DNA long, which means they're really useful for looking using the PCR reaction. So in other words, if we've got small samples, then we can use these particular markers. And I'm going to go back a second because I've forgotten the entire point of this slide. It was not to talk about monogamy. It was to actually talk about the fact that if anyone's done any mammal trapping at all, you will understand the idea of a trap-shy animal or a trap-happy animal. And there are animals that love going into traps and they love that peanut butter bait. And I've got individuals where I would set the trap, I would take 2 steps and you'd hear this and there'd be an animal sitting in the trap. And you'd drag the animal out, send it on its way, rebait it, set the trap, take 2 steps, and the trap went off again. This is what's referred to as a trap-happy animal. They love getting in there and stuffing their faces in that bait. Then there are individuals where we simply never caught them. They were so trap-shy that we never had the opportunity to take a DNA sample from, say, the male from those samples. So we could catch the female, we could look at the young, so we had DNA from the mother, and DNA from the young, but we never had the male. So we could never actually work out, I mean, it would be ideal to have the male as well. So this got, and this is really early on, so this got us into the idea of, well, we need to try and sample these males. How do we do it if we can't actually get them into a trap? Because you can't catch them like it's there as, anyone said a rock wallaby working rats, puts away around a rock, they'll appreciate they're pretty nimble little things. So what we did was we set up mosquito traps. And mosquito traps are a UV tube, they're sort of about this big. They're a UV tube, and they have a fan in them, and then they have a sock out the end of it. Basically sits on a tripod. So what we did was we knew that individual rock wallabies were sitting on individual rocks. Another set of individuals would be sitting on another set of rocks, very close by, but I tell you, if you started to walk in there, they'd start to beat the hell out of you because they're quite protected with their little patch. We set up a mosquito trap on each of these rocks that we wanted and that we knew. So we had behavioural ecologists working out there, looking at these rock wallabies, and they couldn't see ear tags in them. So we had ear tags in them, etc. So we know which ones weren't captured. So we set these mosquito traps down, and then we would sample the mosquitoes out of that trap in the morning. And it was really cool because what we were able to do was to take the blood meal from the mosquitoes that had bitten the rock wallabies and amplify up the DNA from the individuals we never caught using the blood meal using PCR. So there's a classic example of using really small amounts of DNA in my opinion, a really unique way to try and identify individuals we're going to catch. So that was the idea of this slide. Okay. Oops. Sorry, just to go on about microsats. So they're highly polymorphic. They're very small in size, so we can use them for PCR. And the other thing is there are lots and lots of them in the genome. So every 1,000 base pairs out of our 3 billion genome-sized thing, we find some microsats, so they're easy to identify. The disadvantage is we need to identify them for each species. So we need to reinvent the wheel every time we've got a new one. So if we've got numbats, we get new ones. If we've got bootle owls, we've got one. Box jellyfish, we've got a set. Kangaroos, we've got a set that work for them, etc., etc. So it can be quite expensive. This is what they look like. Again, I'm not going to ask a question in the exam. The idea is just to sort of give you a bit of an idea of what these microsats look like. Is you're sequencing along the genome, and it's basically random DNA. And then all of a sudden you hit this highly rich region in the middle here, which is this repeat of a CA, 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 CA. In fact, this is repeated 27 times. This comes from a numbat, and we were able to design PCR primers. So PCR needs 2 primers, one on one side of it, the other one on the other side of it. And then we can amplify this big bit in the middle here. And what happens is that this big bit in the middle here varies in size depending on what individual it is. So you'll inherit 1 repeat from your 1 strand of DNA from your mother that might have 27 repeats, and the strand that comes from your father might have 5 repeats in it. So in other words, there's our variation. You can have different sorts. You can have combinations of AC or GA or TA, and you can also have different sizes of microsatellites as well. There are trinucleotides, etc. Okay, this is another region. This is from a Carnaby's black cockatoo. So we did some work on Carnaby's black cockatoo. To the extent we're able to identify every individual from the Carnaby's population. In fact, if you go and nick a Carnaby's black cockatoo out of a tree hollow, good chances are we'll be able to tell you which tree you've taken it from, because we can go and DNA profile that seized bird and identify what tree it came from. We've got more mapped out. So it's very cool technology. But this comes from a Carnaby's, and this is an example of what they refer to as a trinucleotide. So rather than having CA, this one here's got AAG, 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 etc., etc. Just a different sort of microsatellite. And then you've got even bigger ones that have 4 repeats, and these are all the ones used for human forensic profiling. So they're the ones that sit in the DNA kits they're using for, say, they're on serial killing at the moment. So how do we actually get this genetic variation? We look at these different markers and whether there are any variation in there, and we can see these are 4 different genetic markers. One's in black, one's in blue, there's a green one and there's a red one. And if you look at the one in blue, for instance, you can see that these are 3 different individuals here. And it doesn't matter which individual you look at, they all just have that 145 allele in there. So it's actually no use at all in conservation genetics for looking at diversity, because they're referred to as monomorphic, they're all the same. What we are after is variation. And you can see in this black one, there's one individual here that has an allele 129. And the way you would in fact score it would be 129 and 129, because they have 2 copies of it, they just happen to be the same size. This will be very obvious this afternoon, so don't worry too much about this. But you can see this individual's got a 129 allele, this one here's got a 131 allele, so there's already variation in there. And this individual is heterozygote, meaning it has the 2 different alleles but in different sizes, so it's a 129, 131. And the same thing goes for all these ones here. So there's a reasonable amount of variation in these systems, and we can work out the frequency with which these alleles occur in populations. Right, so I'm going to sort of move on from the technology more to how we apply this stuff. Is the greater the number of different alleles, so the more we have of these alleles in a given marker, then the greater the number of possible genotypes. In other words, there are genetically different individuals in the population, and that's kind of what we're trying to aim for. And as I said before, there's general consensus amongst population geneticists that the higher the heterozygosity, or the higher the genetic variation, the more the population has the capacity and ability to buffer change, and that's what you're actually interested in. So I'll give you an example where we've used these 3 different types of technology on the same beastie, and I'm going to come back to these allied rock wallabies, where we published a paper where we took a group of individual rock wallabies and we measured genetic variation using alizyme markers, using multi-locus DNA fingerprinting markers, and again, I'm not expecting you to understand exactly what I mean by these markers, but just there are different types of ones. And then we measured microsatellites, these highly variable ones. And again, don't worry too much about the details of all these. I just want to look at a couple of things. One is, I want to look at heterozygosity, and heterozygosity is just looking at how heterozygous, how different those individual alleles are in individuals. When we used alizymes, there was about 17% genetic variation. When we used DNA fingerprinting, there's about 50% genetic variation. And when we use microsatellites, there's about 85% genetic variation. What I'm not saying is, I'm not saying that using different markers gives you very different results. In other words, rock wallabies are really variable, or they're not actually really variable. I'm saying that the markers are variable in those populations. So if we want to look at and compare different populations, we are much better off using microsatellite markers, because we have a lot more variation in there to do comparisons with than we are to do with alizymes, where there's not as much variation. If we look at the number of alleles per locus, in other words, we take genetic marker number 1, and we look at the population of rock wallabies that we've got, and then we look at how many alleles are in that population, there's only about 2 or 3 alleles in any population of rock wallabies using alizymes. But if we use microsatellites, then there's 11, 12 alleles in any population. So there's a huge amount, or a lot more variation in using microsatellite markers than there are some of this older technology, which is why we're using microsatellites. Does that sort of make sense now, Greg? I think I'm starting to lose everyone. We'll keep going. There are different ways of measuring genetic variation. So we can use a number of different genetic measures looking at variability. We can look at the number of polymorphic genes, we can look at the number of alleles, we can look at average heterozygosity, etc., etc. So just to show you that there are different things. So this will give you a bit of an idea about what I'm talking about. Has everyone heard about Hardy-Weinberg equilibrium? Yeah. Hardy-Weinberg equilibrium, if you've sort of forgotten, is an ideal population that describes a population that is infinitely large, where every individual could potentially randomly mate with any other individual in that population, where there's no immigration, there's no migration, there's no mutation, etc., etc. So in other words, it's describing something that's absolutely ideal. The reality is they don't exist. But this is how Hardy-Weinberg sort of works. And I've got this sort of idea of this Hardy-Weinberg island. And what you've got is you need to imagine that there's an infinite number of different coloured dots in that population. We have no random mating, we have an infinitely large population, no selection, no migration, etc., etc. So if we look at that same population, 50 generations later, what we'd expect to see is basically what we found originally. So everything's randomly mating, so we'd still have an equal chance of finding a yellow, or a blue, or a purple, or a red, or a green, etc., individual in that population, because everything's behaving ideally. What invariably happens is we have things like vicariant events. Vicariant meaning something like a mountain range comes up between two populations, all of a sudden the population splits into two. So we have a west and we have an east population, and that island becomes divided such that individuals from this side can't actually mate with individuals from that side. And this happens all the time in populations. So if we go forward one generation, the population should theoretically look roughly what it was before, because there hasn't been a lot of time between those changes. If we look at 10 generations, we'd expect to start to see small differences between them, where we've got, where we have more, if we were to sample this side of the population, the western side, more of those representations of the yellow ones than we had previously. If we jump forward 30 generations, we have increasing differences between them. If we go up to 50 generations, we're starting to get to the stage where we've got quite large differences. And at some stage, at some point in time, those populations are going to become fixed. Individuals that were purple will ultimately get purged from the population because they're simply undersampled, and the populations will be divided into essentially two different populations that are genetically quite unique from one another. There's a yellow group here and a blue group here. What would happen if we still assumed Hardy-Weinberg in that population? It would fall apart because we're no longer in Hardy-Weinberg. In other words, we can look at a population and look at the distribution of the alleles or the genes in those populations and infer something that's changed the population. In other words, there's something that is not making that population randomly mating, infinitely large, where there's no immigration, no migration, etc., etc. Does that make sense to everyone? So you can actually use genetics as an indicator of where things are starting to go wrong in populations. And this is what I want to spend a little bit of time on in the next little while. So what happens in reality is, in fact, the population would not be in Hardy-Weinberg, and we would calculate different databases depending on whether we were from the east or the west side of the population. So imagine the old boundaries got forgotten and all of a sudden there's happy mixing and individuals in the population. What was effectively two distinct populations would start to do what they were originally doing, where you have individuals mixing. So individuals moving from the west to the east to the west, etc., etc. And the other thing that would happen, of course, is that some rare mutations would occur eventually in the population. So we get new individuals occurring in these populations as they're starting to mix. And you would maybe expect individuals to move from different populations back into those populations. In other words, the population's starting to behave itself again. Ultimately, what we would expect is the populations would then come back to what we originally had, which was a genetically homogeneous, very large, randomly mating population with no natural selection, no mutation, etc., etc., etc. It's important to understand these things because if we see a population that's changing, we can infer the reason why they're changing and do something about it. For instance, if we fragment a population and there's no migration into that population, it may be in a national park or a wildlife reserve, then obviously one of the mitigating management options might be to take an individual or group of individuals from somewhere else and artificially put them into that enclosure. In other words, reinvent migration into that population by translocating intervals. So there's a management option you could use to try and ensure that the gene pool increases. So for management purposes, if we know what causes the changes or the disturbances in genetic diversity, then we can mitigate against them. So in other words, if we're looking at mutation, the primary cause of all evolution, mutations are the reason we get new genetic information. It is something that we really can't do a lot about. So we can't form new organisms. It has to happen over time, and it happens very slowly. If we look at genetic drift, then it's basically the loss by chance of alleles in a population. So you take a big population, you fragment it, and this population only has those yellow plus a couple of other different variations in genes, then very quickly you will probably lose those small frequency alleles and you're left with this one homogeneous population of a yellow colour, for instance. Gene flow, very important for distributing genes amongst populations. And if you think about it, it's the opposite of genetic drift. So if you have two populations where you have the two different colours, the yellows and the blues, and all of a sudden you introduce migration, then you very efficiently and very effectively remix those genes in that population as a result of individuals moving between them. Non-random mating. Basically, one of the assumptions of Hardy-Weinberg is that everything is randomly mating. In other words, there's an equal chance of any individual in that population mating with any other individual, reassorting those genes in the population. The reality is that just doesn't happen, and I'm going to give you some extreme examples of that in a second, where you have really unequal distribution of mating. So if you've got one dominant male, say in the big kangaroos, the greys and the red kangaroos have a very hierarchical male dominant system where you've got one very large male that dominates essentially a group of females. There's only his genes represented in those further generations. It's not a randomly mating population. It's dominated by a single male. Okay. So, and the other one is changes in population size, and this really affects a lot of endangered species where you had very large populations randomly mixing, and all of a sudden those populations shrink because of disease, because of predation, fragmentation, etc., etc. What that does is it affects the size of the population, and it's a really important concept to get our heads around. So I want to spend a little bit of time talking about population size and try to give you some examples where it sort of hopefully makes sense as to why it's so important. So population size should be a good estimate of the number of individuals in a population, and therefore, by default, should be a relatively good indication of the genetic diversity. The more individuals in a population, the more genetically diverse they should be. Make sense? It's referred to as, and we would base this population size estimate on what they refer to as the census size. So we go to Rottnest Island and we count 10,000 hoppers on Rottnest Island and sit there and they go, these are fabulous, they're doing really well, there's bucket loads of them. In fact, there's more than bucket loads because you simply can't even cope with the number of individuals. But the problem is census size is actually a very poor indicator of the absolute population size for a whole range of different reasons. There are individuals in that population that are too old, that are too young to reproduce, that are infirm, that are dominant, or in fact that aren't actually reproducing. So subdominant males won't actually be introducing any of their genes into subsequent populations. So if your population is comprised of one dominant male and 100 subdominant non-reproductive males, then those 99 are not being represented in the next generation, it's just that one. So that census size becomes quite biased and towards that one individual. So this is where conservation genetics introduces this idea of what they refer to as effective population size. And effective population size are those individuals that are effectively contributing to the next generation. In other words, that one dominant male would be important in estimating effective population size because effectively they're introducing them. So effective population size usually contributed effectively by a few individuals, and as such, the census size may actually be a really poor indicator of the actual size of the population. And I'll give you the heads up with Rottnest Island. There's 10,000 individuals in that population. The effective population size at Rottnest Island is about 100. So there's effectively 100 contributing quokkas on Rottnest Island. The other 990,000, get the numbers right, the other 9,990 or 900 individuals in that population are not effectively introducing any new genetic material in the population. So the vast majority of Rottnest Island quokkas are effectively clones and not contributing anything to future generations. It's actually a very small population in terms of effective population size. So 10,000 individuals sitting on Rottnest Island, 100 are actually effectively comprising of the population from Rottnest Island. I'll show you some numbers in a second. An effective population size is really important because what it actually is it's determining the rate of loss of heterozygosity in each generation. So it's a really good measure of how populations are losing diversity. And ME is also important because it determines not only the loss of heterozygosity, but it's influenced by a whole range of different things. And I'll talk about a number of these things. The number of male and female individuals in a population, the mating system. Is it a hierarchical system? Is it a random system? Is it a monogamous system? The variation in family size. In other words, those individuals in a population, and these rock wallabies are really interesting. The reproductive success spans an enormous range in those rock wallabies. You've got some individuals where every single young they produced was raised successfully. And you've got other individuals, and I did this stuff 20 years ago, there's rock wallaby 133. This poor female endlessly produced rock wallaby young, and not a single one of them ever survived to independence. Every single one of them died. So you've got females that are producing very low variation in terms of reproductive output. So variation in reproductive output affects effective population size, as does fluctuating population size itself. So you've got some individuals, for instance, humpback whales, a classic example, numbers in hundreds of thousands prior to whaling, reduced down to, I'm going to make the number up, it's something like 10,000 individuals or something at the end of whaling, and all of a sudden they're gone nuts, they're back at 100,000 I think at the moment. So that's a huge fluctuation in population size. Big populations have been really reduced and then come back out of it. And there's also generational effects. So what I want to do is to talk about each one of these and the contribution that each of them make to effective population size and population. So just a few definitions. It's an ideal population in which every individual has an equal probability of contributing to subsequent generations. That's what effective population size is. It's a very theoretical, abstract term. So in other words, don't take absolutes at, it's got an effective population of 7.2. What you're really interested in is looking at comparisons. This population has an effective population size of 10, and this individual population size has effective population size of, say, 100. So this one here is effectively 10 times bigger than this population here, rather than absolute numbers. I think it's a bit weird. And there are two terms. One is census size, and the other one's effective population. So this is how many you'd actually count if you actually went out there. And this is estimated using genetic properties in the same population. Oops. Okay, so number of males and females. Not all individuals in a population contribute gametes to the next generation. Now we know that a lot about that. And there's a relationship between them, and this is the relationship. And again, if I was to ever ask you in an exam, I'm not going to ask you to produce that equation. What I would be asking you is, can you understand what the effect of the number of males and females have to subsequent generations in these things? So there's a relationship between effective population size and the number of males and number of females in a population that are actually breeding. And the effective population size is nearly always much smaller than the census size. And as a general rule, there's a really good friend of mine called Dick Franklin who's written some fabulous papers looking and quantifying the metrics on those. And generally speaking, the effective population size is about 1/10 of the size of the census population. So every time we go into a population of koalas and we count 100 koalas in there, the effective population size is usually about 10 or so in the population. So I'll give you an example with elephant seals. Everyone's aware of the elephant seals. These are huge things. I've run into one once and it was sort of about that big compared to them. So they they're an enormous animal. But they have a very, very typical male hierarchical system. There were about 100,000 elephant seals prior to them harvesting them for their oil, and they were reduced down to 20 or 30 individuals by the mid-1800s. So they went through a dramatic population decline. They're about 100,000 now, so they've recovered quite nicely. but there's absolutely no detectable protein allozyme variation in that population. We can't detect any alleles that are variable between individuals. These are looking at 24 different markers, so it's a large amount of information. If we assume there are 500 adults, there's a 50-50 sex ratio, which appears to be the case in the population, and there's equal reproductive success, then if we do the calculation based on that, then the effective population size is actually very close to the actual census size. In other words, if everything is behaving properly, the effective population size is very similar to the census population size. Does that make sense? That's a good thing. But the reality is that it doesn't have this situation. What we have is a situation where we have a dominant male with a social structure that excludes subdominant males from mating with females. So if we have a harem of females with a single male that breeds, say, with 100 different females in that group or that harem, and we do the maths the same way, effectively the effective population size works out to be about 4. In other words, we're no longer, the effective population size is equal to the census size. It's actually a very, very small number compared to the actual census size of the population. So it's only about 4% of the actual size of 100 individuals. If we take 100 plus 1 male, the census size will be 101, and the effective population size is very much smaller because of that social hierarchical dominant system that happens. So the sex ratio has a very profound effect on effective population size. If everything being equal, and everyone's randomly mating and everyone's happy, then that census size approximates the actual effective population size. If we look at the variance in family size, and we're talking about this when we're talking about where some females produce lots of young, and there are other females that really don't have very many young at all. So in other words, we're looking at the variation in there. So this is the census population size. We're keeping that at 100. And if we look at a population size of 100 where the variance is very small, in other words, there's lots of individuals producing lots of young, then the effective population size is relatively large compared to census size. But as there's a large amount of variation in there, as the variance in reproductive success comes into play, then the population decreases dramatically. In other words, if you were looking at, and you had to do the maths, then if you had a variance of 10 with a starting census size of 100, the effective population size would only be about a third of the actual census size. So it doesn't affect it as much as the number of male to female, but it certainly has an effect on the effective size of the population. And the last one's looking at fluctuations in population size. And this is an example, this is a classic example of the snow and the hare. So basically you have, they've done a whole lot of work, I'm sure you would have seen this in ecology, where you have the number of rabbits increase, and lag behind that will be the number of lynx or the number of predators that are associated with eating. The rabbits then decline, therefore the predators decline, and it goes through this cycle of up and down through time. And these are from some really nice, really elegant ecological work looking at track records. The effective population size in those fluctuating populations is determined using this equation. Again, I'm not going to go into the equation, I'll give you the equation, but the idea is you need to understand what the equation is telling you. Is if you can assume that initial population size is 100,000, it then went down to 20, like it did with elephant seals, and then it moved back to 100,000 subsequently. In other words, the population is very large, it's reduced dramatically, and then it's back at 100,000. You go to that population, you count, there's 100,000 elephant seals sitting on that beach. But the reality is, again, if you do the mathematics of 5 generations, and you chuck the numbers in terms of the maths of the effective population size, 100,000 individuals lying on a beach sunbathing, but when you actually look at it, the contribution of effective individuals is closer to 300, and it's much closer to the smallest population size than it is to the existing 100,000 census population size. Does that make sense? So the fluctuations, even though there's 100,000 individuals in a population, it's much more likely to be much closer to the smallest population size that it's ever been than it is to any of the large population size. So fluctuation in population size, and this is really important in conservation because we've seen populations There's some really elegant studies by Jeff Short up from Shark Bay, and looking at the effects of cyclones and environmental characters on some of the Shark Bay heron animals, and they show exactly that. These populations just fluctuate year in, year out in terms of survivability, population size, etc. So fluctuating population size is super important. And the other ones, when we look at what happens as a result of the loss of variability, this is a really scruffy graph, but if we look at the amount of variance remaining, in other words, the amount of heterozygosity remaining in a population, and we look at this over time, if we have a very large population, then we lose very little variation over time. But it doesn't take too long till those populations, when they become smaller and smaller and smaller, that we start to lose variation very quickly. So in other words, large populations retain diversity, and as you go down in population size, this is a population size of 10, is after 10 generations, it only contains about 70% of the variation that it had 10 generations previously. If we take one individual, population size of 1, then within 10 generations, effectively no genetic diversity left in that individual or population as a result of loss of diversity. Make sense? So the gist of this is large populations can buffer those changes. Small populations are very, very dramatically and very quickly affected by loss of diversity. So if we were to look at a population with assumed 10 generations, an effective population size of 10, then after those 10 generations, there's only about 60% of the diversity remaining after that time. So we really want populations, if we're trying to manage them in terms of conservation reserves, nature reserves, to have as large a population as we possibly can get. And in fact, there's even numbers associated with these. 500 seems to be the magic number at the moment, changes periodically. But they say that 500 is sufficient to maintain genetic diversity in any endangered or threatened species. The reality is a lot of these species don't actually have 10. I think western ground parrots have now gone down to 10 individuals. Been involved with the early stuff with that. This has just been a tragedy, those guys. Anyway, the point of it is that how many individuals you see in a population can be quite deceptive, and they're influenced by a whole lot of things, and they're quantified using this measure called effective population size. And we'll talk a little bit about that later on as we go through this thing. And this is one of the results of very small effective population size. This is a quokka from Rottnest Island. These are the sorts of things that happen when you have very small genetic population. Anyone see it? Anyone not see it? Yeah, it's got 3 years. These are the sorts of things people talk about, you know, when you... Anyone? Oh, no, I won't say it. Okay. The second area I want to talk about is called genetic bottlenecks or founder effects. And these are, again, really important in conservation planning, because this is exactly what happens when species that have very large distributions and very large numbers of individuals shrink down dramatically, and these are the consequences of doing so. These are really sampling effects in a population. This is where you have a population of 100,000 individuals and they get hunted down to 50. Is those 50 that are remaining will be independent of adaptation. They're not going to worry about whether it's big, small, highly fecund, whatever individuals. It will just be randomly removing most of those individuals from the population. And so I just want to talk about this. So this is this effect called the founder effect. And there's some just nice examples, so I'll just rip these off to try and make an example. So you can imagine that you have the original population, lots of different alleles, lots of different colours, these marbles representing different alleles in that population. And if you reduce that population due to hunting, disease, fragmentation, whatever you have, then only a few individuals will go through the bottleneck in that population and be available to reproduce in subsequent generations. So what you had effectively were a large genetically diverse population now becomes a very small genetic pool in which subsequent generations can reproduce from. Those generations that are the result of that bottleneck will only comprise of individuals that were in the founding populations. The diversity that were in the original populations is simply lost, no longer there. And as I said, a genetic bottleneck creates random genetic changes in populations without any consideration for adaptation. These individuals that are surviving are not necessarily the most fecund, they're not necessarily the largest ones, they're not necessarily the ones that are more climate adapted or whatever. These are simply a random effect. It's basically the same as grabbing a bag of marbles, putting a hand in, and grabbing a handful, and that's the remaining individuals. They could be the village idiots, they could be the village slobs, they could be the people, the ones that don't actually do anything, etc. You know what I mean? So they're not adapted individuals. I'll give you an example, Australian examples. So anyone coming this afternoon will meet Dr. Darren Alpers, and Darren did her PhD on southern hairy-nosed wombats, and another friend of mine, Andrea Taylor, did her PhD on northern hairy-nosed wombats. Everyone heard of hairy-nosed wombats? They're really big, so pick one up, and they're, you know, if they grab them under the arms, they'd be about from here to the ground, so they're quite a large animal. And they used to occur from sort of mid-Queensland, inland Queensland, all the way around through to Denali and down towards the Victorian border. So they had a very large distribution in the northern area of Bottleneck. They now live on a reserve, and that reserve is about the size of Murdoch Uni campus, South Street here. So it's a really small area, and the frightening thing is looking at the aerial photographs where you've got this island of bush, and you've got this completely cleared pasture around this reserve. So there's just no capacity for animals to move anywhere. And when they looked at these hairy-nosed wombats, they compared them using, again, these microsatellite markers. There's 16 of them, so there's a large number of genetic markers they used. And when they looked at the southern hairy-nosed wombats, these things contained between 3 and 4 alleles per locus in the southern hairy-nosed. They're not threatened, they're not endangered, they're doing quite well. And heterozygosity of around about 65% or so. When they did exactly the same markers on the northern hairy-nosed wombats, there was only between about 1 or 2 alleles per marker on average in northern hairy-nosed, and the heterozygosity was reduced to about 30%. So these northern hairy-nosed wombats have been through this very large bottleneck, where there was a very large population, now reduced to a couple of hundred individuals living in this nature reserve. This is another example, dibblers. Hopefully we might see these if we go to the zoo. And basically, they've got an interesting history. They've got, dibblers were, they thought they were extinct, and then in the mid-60s, they found them in this area around the Fitzgerald National Park, Fitzgerald River National Park down in the southwest. So there's a mainland population that are doing relatively well. And then they found them on 2 islands off Jurien Bay, one on Boolinger Island, the other on Whitlock. And when we looked at the genetic signatures of these populations, what we found was, if we were to look at the Fitzgerald River National Park, these are the allele frequency classes, and what this figure is showing is, these are the really common alleles down here, and these are the really rare alleles that you only find infrequently in individuals. What it's showing is the mainland population had lots and lots of really rare alleles, lots of genetic treasure in that population. But the populations in Boolinger Island and Whitlock Island predominantly had very common alleles, but they lost all that really rare alleles in those populations. So in other words, they've been through and they show signs of genetic bottlenecking as a result of that. We can use mathematical models to try and actually quantify the amount of bottlenecking they've been through, which is what we did. So there's a West Australian example. And I'll give you another example, turning on its head a little bit. This is the work we did on feral pigs. And if you think about it, if you've got an endangered species, and it's got a very large distribution, lots of diversity and no bottleneck, that's a good thing, because it means the population has lots of diversity in it. If the population crashes like they've done on those islands, then you lose that diversity and you can pick up signatures of genetic bottleneck. In other words, the population's crashed, and that's a bad thing in endangered species. Feral animals is exactly the opposite. If you were looking at feral pigs and you were to look at the diversity and the rarity of alleles in the feral pig population out of Collie. Anyone from Collie down that way? I wouldn't have put my hand up, but anyway. Collie's an interesting area. It's sort of like feral pig central. Like there are some, and I'm talking, the pigs are fine. It's the humans that are trying to just digging myself a grave, aren't I? The Collie's a really interesting area. It's just got this amazingly large feral pig population. I think predominantly preserved or maintained because of the forest system in there. You know, they've got, it's a very difficult country to get into. There's not a lot of people living there, etc. The pigs and collies show no sign of genetic bottlenecking. In fact, they're genetically healthy. Lots and lots of these really rare alleles in those feral pig populations. If we were to look at the feral pig populations from Denbarker, then it's quite different. What Denbarker's happened is there's a whole lot of community groups down there, farmers, DVCA, Parks and Wildlife, landowners, community groups, etc., involved in feral pig control in the Denbarker area. And when we sampled feral pigs from Denbarker, they had lost the signatures of these really rare alleles, and they were having lots and lots more common alleles. In other words, statistically, they have been through a genetic bottleneck. And we actually said to them, we think your population has crashed. And the next year they caught 6 pigs, whereas previous years they were catching 200 something. So if you think about it, a genetic bottleneck in a feral pest population is actually a really good thing, and a real indicator that they're actually having an effect on the decline of that population. So the population control is working really well. Just to give you an alternate view on these things. So, what happens with genetic bottlenecks and founder effects is that if we have a very large population and we were to halve the population and only 50% survive, then the allele frequency would change from about 10% down through to 9%. So we've lost very little variation in terms of subsequent generation, even though we've halved the population size. So large populations can buffer and have small effects in those populations. If we take the same scenario where we've got a population of 10 individuals, then we have a frequency of 10%, same as over here. But if we halve that population, we effectively lose all the diversity in that population. So the take-home message being small populations are affected very, very prominently by small population changes. So small populations are affected and their effects are very large in those populations. It makes a lot of sense. You want big populations, you don't want small fragmented populations that start losing diversity really quickly. Now, we'll have a break for a bit. Okay, we'll have a break for 5 or so minutes and then I'll keep going. Oh, where are some of those things?